Agenda for today, class. What is a drug? Sources of drugs with an examples. The process of drug development. Drug discovery and development. Preclinical research. Clinical research. Regulatory approval and launch. Post-market monitoring. Timestamp links available in description. What is a drug? A drug is any substance that causes a change in an organism's physiology or psychology when consumed. Drugs can be used for medical, recreational, or other purposes, and they can have beneficial or harmful effects depending on how they are used. Types of drugs. Medicinal drugs, used to treat, prevent, or diagnose diseases. Example: antibiotics, painkillers, antidepressants. Recreational drugs, used for pleasure or altered states of consciousness. Example: alcohol, nicotine, cannabis. Illicit drugs, illegal substances that can have harmful effects. Example: heroin, cocaine, methamphetamine. Psychoactive drugs affect the brain and nervous system, altering mood, perception, or behavior. Example: caffeine, LSD, MDMA. Performance-enhancing drugs used to boost physical or cognitive performance. Example: steroids, nootropics. Drugs can be legal. Example: prescribed medications, caffeine, etc. Illegal. Example: heroin, cocaine, etc. And their effects depend on factors like dosage, individual physiology, and method of use. Medicinal drugs. Drug defined as per FDA, a substance recognized by an official pharmacopoeia or formulary. A substance intended for use in the diagnosis, cure, mitigation, treatment, or prevention of disease. A substance other than food intended to affect the structure or any function of the body. Drug as per WHO. Any preparation for human or veterinary use that is intended to modify or explore physiological systems or pathological states for the benefit of the recipient. Conclusion: a drug is a substance other than food recognized by an official pharmacopoeia or formulary intended to affect the structure or function of the body for diagnosing, curing, mitigating, treating, or preventing disease for the recipient's benefit. Sources of drugs with an examples. Drugs originate from various natural and synthetic sources. Understanding their sources is crucial for drug discovery and development. Classification based on origin: natural, semi-synthetic, and synthetic drugs. Plant sources. Many drugs are derived from plants due to their bioactive compounds. Examples: morphine from Papaver somniferum, used for opium poppy. Atropine from Atropa belladonna, used for deadly nightshade. Quinine from cinchona bark, used for malaria. Animal sources. Some drugs are extracted from animal tissues and secretions. Examples: insulin, originally extracted from pig and cow pancreas. Heparin, derived from pig intestines, used as an anticoagulant. Thyroid hormones, from animal thyroid glands. Microbial sources. Microorganisms, especially bacteria and fungi, produce antibiotics and other bioactive compounds. Examples: penicillin from Penicillium mold. Streptomycin from Streptomyces griseus. Erythromycin from Saccharopolyspora erythraean. Mineral sources. Certain minerals are used in medicine for therapeutic purposes. Examples: iron used to treat anemia. Magnesium sulfate, used as a laxative and anticonvulsant. Lithium, used in bipolar disorder treatment. Marine sources. Marine organisms are emerging as important sources of novel drugs. Examples: ziconotide from Conus magus, cone snail venom, used as a painkiller. Cytarabine from marine sponge, Tethys, used in chemotherapy. Trabectedin from Actinicidia turbinata, sea squirt, anti-cancer drug. Synthetic drugs. Entirely made in laboratories to mimic or improve natural compounds. Examples: aspirin based on salicylic acid from willow bark. Ibuprofen, developed synthetically for pain relief. Propranolol synthetic beta blocker for heart conditions. Semi-synthetic drugs. Derived from natural compounds, but chemically modified to enhance properties. Examples: amoxicillin modified from penicillin. Hydromorphone, modified from morphine. Paclitaxel, Taxol, originally from Taxus brevifolia, Pacificu - now semi-synthetic. Biotechnological sources. Genetic engineering and biotechnology have led to the production of biopharmaceuticals. Examples: recombinant insulin, produced using genetically modified bacteria. Monoclonal antibodies, used in cancer therapy. Gene therapy drugs, to treat genetic disorders. So finally, drugs are derived from natural sources, derived from plants, microbes, animals, and minerals. Semi-synthetic drugs, modified versions of natural compounds. Synthetic drugs, chemically synthesized in laboratories. Biotechnological sources, genetic engineering and biotechnology. The process of drug development. Drug discovery is the process of identifying new candidate medications for treating diseases. It involves several stages, from initial research to clinical trials, before a drug can reach the market. This process is highly regulated to protect public health and ensure that new treatments are both safe and effective. Strict regulations are in place primarily due to public health concerns. A novel drug cannot be taken directly from a laboratory and administered to patients without undergoing rigorous validation and approval by regulatory authorities. Drug regulation plays a crucial role in ensuring the safety, effectiveness, and quality of medicines. It prevents harmful or ineffective drugs from reaching patients and ensures proper testing, manufacturing, and labeling. These regulations help minimize risks and guarantee that medicines work as intended. Additionally, they protect human participants in clinical trials, ensuring ethical and safe testing procedures. Developing a novel drug involves two main stages: drug discovery and drug development. In the first stage, researchers identify potential drug candidates, and in the second stage, these candidates are further developed and tested. Each of these stages is divided into multiple steps, making the entire process lengthy, expensive, and highly complex. Drug discovery is not a straightforward task that can be completed within a year or two. It requires immense effort, and even after years of work, there is no guarantee that the drug will reach the market. A potential drug may show promising results in animal studies, but fail in human trials due to lack of effectiveness or unexpected toxicity. At any stage, unforeseen issues can arise, making the outcome uncertain. Researchers invest a significant amount of time, money, and resources into a single drug candidate, knowing that many will not receive approval. The entire process is far more intricate than it may seem, requiring rigorous testing and validation. Now, let's explore the process in more depth. Drug discovery. Target identification. Target validation. Hit identification, screening for compounds. Lead optimization. This process will take 3 to 5 years. The first target identification. Now, before you even think of discovering a new drug, you will obviously think of the disease, right? What actually causes a disease? Scientists identify a biological target example, a protein, gene, or receptor linked to a disease. Maybe it's a genetic disease. So you will come up with the conclusion saying, okay, this particular gene, which is defective, might be the cause of the disease. Or this protein, which might be the probable cause of the disease, right? So you identify the target. Target is any gene or protein which is involved in the disease and which causes the disease. So that is the first step, target identification. Second step, target validation, is the process of confirming that a biological molecule, such as a protein or gene, is directly involved in a disease and can be effectively targeted by a drug. Now, suppose you are studying a disease, you have shortlisted, let's say, 5 genes for a particular disease. Maybe out of 5, only 1 gene is actually involved in the disease. The rest 4 genes are not even closely related to the disease, maybe. So, you need to validate whatever targets you have shortlisted that are actually involved in the disease. At the same time, you check for the drugability. Now, drugability is a term wherein you check the binding capacity to the drug. The same thing needs to be done beforehand, because if it is not binding, it will not give you the desired effect to maybe stop the disease, or to reverse the disease, or to just reduce the intensity of the disease. So binding is a very important aspect to check in the first stage itself. This step ensures that modifying the target will have the desired therapeutic effect without causing harmful side effects. Methods used for target validation include genetic studies, biochemical assays, and animal models. Proper validation increases the chances of developing successful drugs. Next step, lead identification. Now let's say there is a library which has many compounds in it. Which lead is actually binding successfully to the target of yours? If it is binding, you can go further with the lead. Lead compound is any substance which has the greatest potential to bind to the identified target successfully. Lead identification is a key step in drug discovery where scientists find promising chemical compounds leads that can interact with a biological target to treat a disease. It involves screening large libraries of compounds using computer modeling. Simple. Next step, lead optimization. Now, once you have shortlisted the lead compound, you optimize it. Maybe that particular structure is not safe enough. Maybe the efficacy is not good enough. So those certain things are studied, accordingly optimize chemical structures to improve their effectiveness and safety. Once a potential lead is identified, it undergoes further testing and refinement before moving to the next stage of drug development. Drug candidate selection is the process of identifying the most promising compound from a pool of potential drug molecules before entering clinical trials. This step is crucial in drug discovery to ensure safety, efficacy, and manufacturability. This process will take 3 to 5 years. Next, preclinical research. Phase 0. Research. On animals tested in vitro, cell cultures, and in vivo, animal models to assess safety and efficacy. Purpose. Pharmacokinetics, PK, and pharmacodynamics, PD, and ADME, toxicology testing. It will take 1 to 2 years. Preclinical testing. Preclinical testing is a crucial phase in drug development that occurs before clinical trials in humans. Preclinical research is the stage of drug development where scientists test a new drug in the lab and on animals to evaluate its safety and effectiveness before human trials. This phase helps determine the right dosage, potential side effects, and whether the drug is safe enough to move to clinical trials. In vitro testing, cell and tissue studies. Conducted in petri dishes, test tubes, or cell cultures. Helps to understand the drug's interaction at a cellular level. In vivo testing, animal studies. Involves testing in animals to evaluate the drug's effects in a living organism. Provides data on drug metabolism, efficacy, and toxicity. In preclinical research, safety and efficacy studies are essential steps in drug development, ensuring that a potential therapeutic is both safe and effective before it advances to human trials. These studies occur before clinical trials, before testing in humans. The goal of preclinical safety studies is to identify potential toxic effects and assess the overall risk-benefit profile of a drug candidate. Key aspects include toxicology studies. Identifies potential toxic effects and safe dosage limits. Pharmacokinetics, PK studies. Examines how the drug is absorbed, distributed, metabolized, and excreted. Pharmacodynamics, PD studies. It examines how the drug works in the body, its effects, and the right dose for effectiveness. Purpose of preclinical testing. Preclinical testing aims to evaluate the biological activity and therapeutic potential of a drug. Assess toxicity levels and safety before human trials. Determine pharmacokinetics, absorption, distribution, metabolism, and excretion, ADME. Identify potential side effects or adverse reactions. Establish the appropriate starting dose for human trials. Preclinical testing timeline. The preclinical phase typically lasts 1 to 3 years. It precedes clinical trials, which occur in 3 phases before market approval. The transition from preclinical to clinical development depends on positive results from safety and efficacy tests. Preclinical studies must adhere to strict regulatory guidelines before proceeding to clinical trials. Before human trials begin, Researchers must submit an investigational new drug application to regulatory authorities, including preclinical study data, proposed clinical trial plans, manufacturing information. Now clinical research. Phase 1. First research in human. Small group of healthy volunteers 20 to 100. Purpose to evaluate safety, side effects, and appropriate dosage. Determines how the body processes the drug. Success rate 60% to 70%. Phase 1 study mostly open label. It will take several months. Phase 1 clinical trials are: first time a drug is tested in humans after successful preclinical studies. Typically involves a small group of healthy volunteers. Usually 20 to 100 healthy volunteers or patients, depending on the type of treatment being tested. Objectives of Phase 1 trials. Safety assessment determines how the drug affects the body and identifies potential side effects. Dosage range establishes the safest and most effective dose by starting with a low dose and gradually increasing it. Pharmacokinetics, PK, study how the drug is absorbed, distributed, metabolized, and excreted. Pharmacodynamics, PD, examine how the drug interacts with the body and its biological effects. Tolerance assesses how well participants tolerate different doses. Around 70% of drugs move from Phase 1 to Phase 2, though many fail due to safety concerns or lack of efficacy. Most Phase 1 trials are open label, meaning both researchers and participants know the drug being administered. Typically lasts several months to a year, depending on the drug and study design. Phase 2. Research on larger group 100 to 500 patients with a target condition. Purpose. Accesses the drug's effectiveness and further evaluates safety. Identifies short-term side effects. Success rate 30% to 40%. Most Phase 2 trials are single-blinded. Randomized controlled study. Up to 2 years this phase will happen. Phase 2 clinical trials are a crucial step in the drug development process, focusing on evaluating the effectiveness and safety of a drug or treatment in a larger group of patients than in Phase 1. Involves 100 to 500 patients who have the condition the drug is designed to treat. Objectives of Phase 2 trials are: safety evaluation, continues monitoring for side effects and adverse reactions. Efficacy assessment, determines whether the drug works for the intended condition. Assess the efficacy of the drug for a specific condition. Optimal dosage determination identifies the best dose that provides maximum benefit with minimal side effects. Comparison with a control group often includes a placebo or an existing standard treatment for comparison. Key goals. Establish a preliminary proof of effectiveness in treating the disease. Identify common side effects and refine dosing strategies. Provide data to determine if the drug can proceed to Phase 3 trials, which involve a much larger patient group. Success rate and challenges. Only about 30% to 40% of drugs successfully move to Phase 3. Challenges include unexpected side effects, lack of efficacy, and difficulty in selecting the right patient population. Phase 2 clinical trials are often blinded, typically using a randomized, double-blind, placebo-controlled design. Why are Phase 2 trials blinded? Reduce bias. Blinding helps ensure that neither the participants nor the researchers influence the outcomes based on expectations. Evaluate efficacy accurately. Phase 2 trials assess whether the drug works in patients, so a placebo or standard treatment is often used for comparison. Regulatory requirements. Many regulatory agencies prefer blinded trials to provide stronger, unbiased evidence of a drug's effectiveness. Lasts from several months to 2 years. Phase 3. Research. Large-scale studies 1,000 to 5,000 patients multi-site. Purpose. Confirms effectiveness, monitors side effects, and compares with existing treatments or placebos. Success rate 25% to 30%. Phase 3 clinical trials mostly double-blinded randomized controlled studies. This phase will take time 1 to 4 years. Phase 3 clinical trials are the final and most comprehensive stage of testing before a new drug, vaccine, or treatment receives regulatory approval for public use. These trials are designed to confirm the effectiveness of a treatment, monitor side effects, and compare it to standard treatments or placebos. Large-scale enrollment, 1,000 to 10,000, though some trials may include even more. Diverse population. Participants are selected from different age groups, genders, races, and health conditions to ensure generalizability. The main objectives of Phase 3 trials include efficacy confirmation, determines how well the new drug or treatment works in a large population. Safety monitoring, detects less common or long-term side effects. In Phase 3 clinical trials, side effects are closely monitored as the treatment is tested on a larger population to confirm its safety and effectiveness. These side effects can vary depending on the drug, vaccine, or treatment being studied. Since thousands of participants are involved, rare or long-term side effects may emerge that weren't seen in smaller Phase 1 or 2 trials. Researchers compare side effects in the test group versus a placebo or standard treatment to determine if they are truly drug-related. If severe side effects appear, trials may be paused or modified. Regulators like the FDA, USA, EMA, Europe, and MHRA review the safety data before approval. Comparison with standard treatments assesses if the new treatment is better, equal to, or worse than existing therapies. Optimal dosage and usage guidelines fine-tunes dosage recommendations for various populations. Success rate for these trials 25% to 35%. Phase 3 clinical trials are usually double-blinded and randomized, meaning that neither the participants nor the researchers know who is receiving the experimental drug versus the placebo or standard treatment. This design minimizes bias and ensures that the results are scientifically valid. Why are Phase 3 trials blinded? Ensures objective results. Blinding prevents expectations from influencing the outcomes. Regulatory standards. Agencies like the FDA and EMA require rigorous, unbiased data for drug approval. This phase will take long time, like 1 to 4 years, when compared with Phase 1 and Phase 2. Regulatory approval. Drug manufacturers submit a new drug application, NDA application for regulatory approval. Phase 1, Phase 2, Phase 3 data is typically submitted to authorities like the FDA for USA, EMA for Europe, or CDSCO for India for review. If approved, the drug is manufactured and distributed. Success rate 10% to 20%. There is a chance for approval only 1 in 10 drugs. This process will take 1 to 2 years. The regulatory approval process is the procedure by which governments or regulatory bodies evaluate and approve products, services, or activities to ensure they meet safety, quality, and efficacy standards. This process is common in industries such as pharmaceuticals, medical devices, biotechnology, food, chemicals, and financial services. The regulatory approval process for drugs is a multi-phase process that ensures new pharmaceutical products are safe, effective, and high quality before they reach the market. Drug manufacturers submit a new drug application for regulatory approval. The NDA must include administrative and prescribing information, Phase 1 and 2 and 3 summary of clinical data, efficacy and safety data from clinical trials, non-clinical pharmacology and toxicology data, chemistry, manufacturing, and controls, clinical pharmacology and biopharmaceutics, risk assessment and post-marketing safety considerations, regulatory authorities, such as the Food and Drug Administration for USA, and Japan's Pharmaceuticals and Medical Devices Agency for Japan, European Medicines Agency for Europe, and Central Drugs Standard Control Organization for India. A review team is assigned consisting of medical reviewers, pharmacology and toxicology experts, chemistry and manufacturing specialists, statistical reviewers, conducts an in-depth clinical and non-clinical review. If the drug meets safety, efficacy, and quality standards, then it will be approved. Or they may request additional information and conducts inspections if required. An expert panel may evaluate the drug and make recommendations to the regulatory agency. Success rate for approval just 10% to 20%. There is a chance for approval only 1 in 10 drugs. For this approval process, it will take 1 to 2 years. A commercial drug launch happens after a drug receives regulatory approval. This involves multiple steps to ensure successful market entry, patient access, and compliance. Launching a commercial drug is a complex process that involves regulatory approvals, market access strategies, pricing decisions, and promotional activities. Here's an overview of the key steps. Once regulatory approved, then manufacturing and supply chain. Scaling up production to meet market demand while ensuring quality and regulatory compliance. Scaling up drug production for a commercial launch is a critical step that ensures a reliable supply while maintaining quality and regulatory compliance. Transferring the drug's production process from R&D or pilot-scale manufacturing to a full-scale commercial facility. Ensuring the process is reproducible and scalable without compromising quality or efficiency. Market access and pricing strategy. Securing insurance coverage, reimbursement, and affordability programs is essential for a drug's successful market entry. Conducting studies to demonstrate the drug's value, cost-effectiveness, and impact on patient outcomes. Assessing market dynamics, competitor pricing, and willingness to pay analyses. Considering different pricing models such as value-based pricing, pricing based on clinical benefits and cost savings. Cost-plus pricing, adding a margin over production costs. Reference pricing, aligning with prices of similar drugs in different markets. Medical affairs and training. A well-trained team is essential for the successful launch of a new drug. The medical affairs department plays a key role in ensuring that sales teams, medical representatives, and patient support teams have the scientific knowledge and communication skills needed to engage healthcare professionals and patients effectively. Medical affairs acts as a bridge between clinical research, regulatory teams, and commercial teams, ensuring accurate and ethical communication of drug information. Medical affairs team trains sales teams and healthcare providers on drug benefits, safety, and administration. Distribution and logistics in a commercial drug launch. Ensuring drug availability through wholesalers, pharmacies, hospitals, and other distribution channels is critical for a successful launch. A well-planned supply chain strategy guarantees that the medication reaches patients efficiently while maintaining quality, safety, and regulatory compliance. Pharmaceutical companies choose a distribution model based on the drug type, market demand, and regulatory requirements. The entire process, from identifying a drug candidate and conducting preclinical research to obtaining regulatory approval and launching the drug commercially, typically takes 10 to 15 years and costs approximately $1 to $2 billion. Post-marketing surveillance. Phase 4. Detecting rare or delayed side effects that may not have appeared in earlier phases. Observing how the drug performs in a larger, more diverse population. Identifying potential interactions with other medications and effects on specific patient groups. Success rate 70% to 90%. This phase is open-label study. This phase no specific time period, mostly 1 to 4 years. Phase 4, also known as post-marketing surveillance, is the final phase of clinical trials conducted after a drug or medical device has been approved by regulatory agencies and made available to the public. It primarily focuses on long-term safety, efficacy, and real-world effectiveness. Post-marketing surveillance refers to the ongoing monitoring of pharmaceutical drugs, medical devices, vaccines, and other health products after they have been approved for use and are available on the market. It is a crucial phase in the life cycle of a medical product to ensure its safety, efficacy, and quality under real-world conditions. Purpose of post-marketing surveillance: detect previously unrecognized adverse effects. Identify risk factors associated with specific populations or conditions. Ensure long-term safety and effectiveness of the product. Assess rare or delayed adverse events not observed during clinical trials. Monitor off-label uses and their associated risks. Ensure compliance with regulatory standards. Post-marketing surveillance plays a vital role in ensuring the continued safety and effectiveness of medical products. Regulatory agencies, healthcare professionals, and manufacturers must work together to collect and analyze data to mitigate risks and protect public health. Success rate 70% to 90%. This phase is open-label study. This phase no specific time period, mostly 1 to 4 years. The drug development pipeline begins with the discovery phase, where around 10,000 potential compounds are identified. It then moves to preclinical studies, where 250 compounds are tested in laboratories and on animal models for safety and efficacy. In human clinical trials, the process is divided into 3 phases. Phase 1 tests 10 compounds for safety in humans. Phase 2 evaluates 5 compounds for efficacy and side effects in a larger group, and Phase 3 involves large-scale trials for 2 compounds to confirm effectiveness and monitor adverse reactions. After clinical trials, regulatory submission and approval follows, where 2 compounds are submitted for review, and ultimately, only 1 compound is approved for market release. The drug development process begins with discovery and development, followed by preclinical research to identify potential compounds. During this stage, researchers assess safety and efficacy before moving forward. Clinical trials are conducted to confirm the drug's effectiveness and ensure it meets regulatory standards for approval. Even after a drug is approved and released to the market, post-marketing follow-up is necessary to monitor its long-term effects. This entire process costs between $1 to $2 billion and takes approximately 12 to 17 years.